Hello students, this is Dr. V. Sridevi, Assistant Professor of Zoology, Government City College, Hyderabad. Today we are going to discuss about general characteristics and classification of Porifera. In this chapter, what is Porifera, what are the important characteristics of Porifera and how it is classified. These are the things which we are going to discuss in this topic. The word Porifera mainly refers to the pore bearers or pore bearing species. The name Porifera explains itself that they are pore bearers, hence it is Porifera. Poriferans are first lowest multicellular animals, example sponges. Based on the embryological studies, sponges are probed as animals and are classified into a separate phylum. This phylum includes about 5000 species. The poriferans have a spongy appearance and are therefore called sponges. They are attached to the substratum and do not move. So, they are having spongy appearance, means they looks like a sponge. If you keep a sponge in a water, it will absorb water how much it can absorb. Like that, the sponges also absorb water and withhold it. And they are immobile animals, so that they will they need some base or substratum. They have the ability to absorb and withhold fluids. They were initially regarded as plants because of their color, that is green color, and their symbiotic relationship with algae. Hence, they are regarded as plants initially. Then later, their life cycle and feeding system were discovered. After discovery of life cycle and feeding system of poriferans, they are included in the animal kingdom. Now, coming to characteristics of phylum Porifera. Some of the important characteristics of phylum Porifera are mentioned below. The cells of poriferans are loosely organized, as they are the first cellular organisms. They are mostly found in marine water, only few are found in fresh water. They are either radially symmetrical or asymmetrical. Radial symmetrical means they are when you see organism from the center axis, they will spreading rays like a sun, that means radial symmetry. Or asymmetrical, asymmetrical means unequal parts of the body. Their body is usually cylindrical. The scleroplast secretes spicules, while spongin fibers are secreted by spongioblasts. So, the scleroblasts are the special cells that secretes spicules for their protection. They have no organs in their body, as they are the initial multicellular organisms. The organ system is not developed yet. They depict cellular grade of organization. Cellular grade of organization means it is a multicellular organism. The cells are grouped together, but they function differently. Functionally, they are differentiated. That is called cellular grade of organization. The body comprises numerous pores known as ostia and osculum. The central cavity is called sphingocele or atrium, which opens to the outside through the osculum. They reproduce asexually by budding and fragmentation. Here the reproduction is asexual type by budding or fragmentation. Asexual reproduction means the gametes will not be involved in the reproduction, only somatic cells involved in reproductive part. The nutrition is holozoic. Holozoic means here the heterotrophic type of nutrition takes place. They have neurosensory cells, but there is no specific nervous system. They have some neurosensory cells, but there is no specific nervous system is present here. They have the ability to regenerate the lost parts. They can reproduce their lost body parts. The development is indirect and the cleavage is holoblastic. Here development is indirect means there is a larval stage in their life cycle and the cleavage is holoblastic. Cleavage is nothing but a type of cell division that can occur in a fertilized egg, and that egg will be divided into meroblasts. And holoblastic cleavage means the complete division of a cell or egg, fertilized egg. The exchange of respiratory gases and nitrogenous waste occur by the process of diffusion. So, here the respiration and excretion can be done by the process of diffusion. Now, coming to classification of phylum Porifera. Phylum Porifera is divided into 3 classes based on the type of spicules and skeletal composition. They are Calcarea, Exactinellida and Demospongia. Now, coming to Calcarea. What are Calcareans? What are the characteristics of Calcareans? They are found in marine, shallow and coastal waters. Here marine means the salt waters or sea water. The skeleton is composed of calcareous spicules. That calcareous is made up of calcium carbonate. The body is cylindrical and exhibits radial symmetry. The body organization is asconide, cyconide, leuconide type. These are the 3 types of canal systems found in sponges. Asconide type of canal system is in which the presence of choanocytes lining on spongocele, the central cavity lined by the choanocytes, that is interrupted only by porocytes. Example, Leucosolenia. Cyconide, it is the more advanced than asconide type of canal system. It is having more advanced canal system than the when compared to asconide type, it is more advanced. And example of cyconide canal system, the Scypha. Now, coming to leuconide, in which the canal system is with flagellum, with a flagellated chambers. The example is Spongilla. Calcareans examples Scypha and Clathrina. Now, coming to Hexactinellida. They are found in marine and the deep sea. The skeleton is made up of 6 rayed siliceous spicules. Here the siliceous spicules means they are made up of silicic acid. The body is cylindrical in shape and exhibit radial symmetry like calcareous animals. The canal system is cycon or leucon type. Example, Euplectella and Hyloneuma. Now, coming to Desmospongia. They are found in marine or freshwater. Here some of the organisms can be seen in freshwater also. The body is asymmetrical and cylindrical in shape. Some of the organisms exhibit asymmetrical and some of them showing cylindrical shape of the body. The canal system is a leuconide type. Here the canal system is leuconide type, means the flagellum is present in this canal system. The skeleton comprises spongin fibers, siliceous spicules made of silicic acid, which are monoaxon and triaxon type. For example, spongia and spongilla. Some of the examples of poriferans are cycon, Hyloneuma, Cliona, Euplectella and Spongilla. Now, coming to cycon, these are solitary or colonial marine sponges found in shallow waters attached to the rocks. Cycons, some of them are living in a living alone, or some of them in the form of colonies, grouped as a colony. So, that they are solitary or colonial marine sponges, and they need to attach to some base. Here they are attached to the rocks. The body is cylindrical in shape with numerous pores. The radial canal is made up of flagellated cells. Here the flagellated cells are helpful in expel the water from the body to outside. Water enters the body through ostia and reaches the radial canal by prosopiles. These species undergo both sexual and asexual mode of reproduction. So, cycons can undergo, some species can undergo asexual reproduction and some of them lead sexual reproduction. Gametes can involve in sexual reproduction, that you know. This is the structure of cycon. So, there are several cells which are exist in the epithelium of the cycon. So, the shape of cells are amoeboid shape. So, they are amoebocytes. This is osculum, that is the largest opening that will expel water from the body, that will be used as the excurrent pore. And there are choanocytes which are present, they are also called as collar cells. This is ostium, this is ostium, that is incurrent pore. Ostium means incurrent pore, that will send water to the central cavity of the body of the cycon. And porocyte or pore cell, this is mesohyal, and this is spongocele, the larger cavity is called spongocele and also called as atrium, and paragaster. This is spicule. So, this is the pinacocyte, epidermal cell, and this is ostium, incurrent pore. This is incurrent pore, ostium. Now, coming to microscopic view of a poriferan wall, that is cycon wall. Here you can see the shapes of different cells. See, this is the spicule, it is like a needle shape, and arachnocyte or amoebocyte, like a amoeba. The shape of the cell is like amoeba. So, it is a amoebocyte or archaeocyte, and this is a sclerocyte. Scleroblast cells can secrete spicules. So, this is a sclerocyte. This is mesenchyme. This is mesenchyme, and this is pinacocyte. This is choanocyte. So, there are different type of cells which functions differently, and this is porocyte. This is porocyte, ostium. This is the canal, The sponges are having a canal system, that is the important feature of sponges. So, this is the canal. So, this is flagellum, this is the collar cell, this is collar cell, this is flagellum, this is the interior part of the cell, this is exterior part of the cell. The skeleton of cycon is made up of spicules that arise from the scleroblasts and are organized to safeguard the delicate parts. The skeleton of cycon is made up of spicules, that spicules protects the body parts, delicate parts of the cycon. A common observance is the presence of tetraradiate or triradiate spicules. The spicules are of tetraradiate or triradiate type. The mesenchyme is composed of a gelatinous proteinaceous matrix containing spicules and many amoeboid cells. It has two kinds of cells, pinacocytes and porocytes. Now, coming to Hyloneuma. These are also known as glass roof sponges found in marine water. Marine water means sea water or salt water. The body is round or oval. Small amphidiscs are present in the skeleton. Now, coming to Cliona. They are also known as boring sponges found in coral skeletons, mollusk shells and other calcareous objects. They are green, purple or light yellow in color. The canal system is characteristic of the leuconoid type of sponges and they reproduce asexually and sexually. Now, coming to Euplectella. Euplectella is a sponge, it belongs to the phylum Porifera, found in deep waters. It is also called as Venus flower basket and given as a gift in Japan marriages. The body is cylindrical, long and curved, fastened in the mud at the bottom of the sea. The canal system is simple cyconite type. The skeleton consists of siliceous spicules fused at the tips, forming a three-dimensional network with parietal gaps. Now, coming to Spongilla. They are largely found in ponds, streams, lakes, growing on submerged plants and sticks. The only species, that is Spongilla, which is found in ponds, streams, lakes and freshwater bodies. The body wall consists of a thin dermis provided with pores called ostia. The body wall consists of thin dermis provided with pores called ostia. Earlier we have discussed about the poriferans example sponge, how the sponge is having pores on its surface. The sponge, these Spongilla also have a pores on its surface. They possess a rag-on type of canal system. They reproduce sexually as well as asexually. So, these are the some of the important examples of poriferans. Leucosolenia, this is Euplectella, this is Cycon and this is Hyloneuma. So, all of the organisms are having the larger pore, that is osculum. It will expel water from the body to outside. So, these are all the small pores which are present on the surface of the body of the organisms that send water into the body, that is spongocele. Now, coming to types of cells. There are four important types of cells are there in its body, that is coinocytes, pinacocytes, lophocytes and porocytes. Coinocytes have flagella that propel water through the body through the osculum. Pinacocytes can transform into any cell type. According to the situation, the pinacocyte can be changed into different types of cells to perform that particular function. And lophocytes that secrete collagen. Porocytes control the flow of water through pores in the sponge. They will regulate the water flow. Collar cells or coinocytes are specialized cells that are characteristic of phylum Porifera. These coinocytes are the specialized cells that are important characteristics of the poriferans. Each coinocyte consists of a central flagellum or cilium which is surrounded by a collar of microvilli. The function of coinocytes is to create water current by beating the flagellum or cilium. The food particles in the water are filtered by the microvilli and is digested intracellularly. Now coming to mesenchyme in Porifera. So, what is mesenchyme? What is the function of mesenchyme and how it is developed in poriferans? In some invertebrates such as Porifera, Cnidaria, Ctenophora and some triploblasts, namely the acoelomates. Triploblast means the 3 germ cells will be present in a dermis. So, the term mesenchyme refers to more or less solid but loosely organized tissue that consists of a gel matrix, the mesoglia, with various cellular and fibrous inclusions. Now, coming to mesenchyme in Porifera. In some invertebrates such as Porifera, Cnidaria, Ctenophora and some triploblasts, namely the acoelomates. Acoelomates means there is no coelom present in the organism, and triploblast means there is a presence of 3 layers, germ layers, that is epidermis, endodermis and mesoderm. The term mesenchyme refers to more or less solid but loosely organized tissue that consists of a gel matrix. That is why it is called mesoglia. With various cellular and fibrous inclusions located between the epidermis and dermis. The mesohyl, formerly known as mesenchyme or as mesoglia, is the gelatinous matrix within a sponge. It fills the space between the external pinacoderm and the internal coinoderm. Mesenchyme of Porifera. Mesenchyme is a type of loosely organized animal embryonic connective tissue of undifferentiated cells that gives rise to most tissues such as skin, blood and bone. Mesenchyme, also known as mesohyl in sponges, is a gelatinous matrix that contains skeletal elements and archaeocytes or amoebocytes. Pinacocytes are thin flat epithelial type of cells that cover the exterior and some interior surfaces of most sponges. The differences between epithelial cells and mesenchymal cells. What are the differences in between? Epithelia have the capacity to function as a barrier or in absorption. Conversely, mesenchymal or stromal cells are loosely organized in a 3-dimensional extracellular matrix and comprise connective tissues adjacent to epithelia. Okay. Poriferans are the organisms lowest developed organisms, they are multicellular organisms. Poriferans means pore bearing animals. So, they looks like a sponge. Their shape is cylindrical, the body shape is cylindrical. Some of the species can show radial symmetry, some of them are asymmetrical, and some of the species can show asexual reproduction, and some of them are leading sexual reproduction. Initially they are considered as plants because of their green color and their symbiotic relationship with algae. After discovery of life cycle and feeding system, they are included in the animal kingdom. They have the ability to absorb water and withhold fluids like sponges, and they are having different types of cells on their dermis. So, in them the collar cells are important, or scleroblasts are important. They secrete the spicules. The spicules protect the delicate parts of the sponges. They are mostly found in marine waters, and some of them can be seen in freshwater bodies. Poriferans are lowest multicellular organisms. That is the reason why they are not fully developed, they are not having the fully developed organ system. So, sponges are having the canal system, and canal system is the important characteristic of the poriferans, and this canal system is the sponges are having smaller pores on their body surface. These pores are helping in bring the water to the body, and they are called ostia, and the central cavity of the sponges are called spongocoel, and the larger opening is called osculum, through which the water will be expelled. So, this is about the poriferans. Thank you, sir.